Chapter 3, it turns out, is just such juicy material that I can't help but extend it by one more lecture. And then next week we'll be back to our usual schedule of each chapter taking 1 week. Okay, so next week we'll be on Chapter 4, the following week Chapter 5, etc. What I want to talk to you about today is DNA reactivity with small molecules, DNA biotechnology, and then how this all impacts and hinges upon cancer. And then if we have time, we'll be on Chapter 4, which is RNA. Okay, so this topic of cancer is something that actually affects, I think, everyone in this room. I think all of us at some point or another know someone who has unfortunately dealt with this terrible disease. It also impacts my students. It impacts students in this room. Every couple of years, I'll have a student who tells me that they fought off cancer at some point in their lives. So it's one of those terrible diseases that strikes all too often. Anyway, our goal is to take a 10,000-foot view, high-level view, at the biology cancer, and then zoom down and start to understand some of the treatments that are used to treat cancer. Okay, some office hour announcements. All right, I have to make my office hour a little earlier because I have to catch a flight to San Francisco tomorrow. And so my office hour is going to be at 1:45 to 2:45. And then my Thursday office hour is going to be canceled. Right, and I assume no one is interested in talking to me after the midterm. I know well how that works. So, okay, so those are the office hours. Let's see what else is happening. Krithika has her office hour today, and Miriam, I think you had your office hour on Friday? Okay. Who knows? Maybe someone will want to chat. I'm sure they want to chat with you all. Okay, anyway, so that's the plan. I'd also like Krithika and Miriam to plan for 1 extra office hour this week before the midterm. So, okay, guys. Okay, so they very graciously agreed. Thank you, guys. Yeah, so you will get an email announcing additional office hours as well. Okay, but I will have an office hour tomorrow at a little bit of an extraordinary time, and then after that, the following week, we'll be back to our usual times. Okay, announcements. Of course, we've already done this. Okay, midterm 1. That's coming up in 2 days, so it's striking really soon. First, there's going to be a review session by the TAs, and the review session, like the midterm, is going to cover through today's lecture. Okay, so everything that's on today's lecture or from the very first lecture is fair game for the midterm. Okay, and if you want to know what's going to be on the midterm, focus on the lectures. Okay, that's what I'll use to drive the midterm as I write it. The seating will be assigned, and it's already posted. Okay, great. So this has already been posted to the website. It's essential that you sit in your correct seats. The seating will be checked, and we'll be checking IDs at the same time. The way this will work is halfway through the midterm, the TAs will make an announcement to pass your IDs to either the right or the left, and then they'll walk around and check the IDs and make sure that everyone's seated in the correct seats. Okay, so you also need to bring a UCI student ID. Now I realize some of you don't have your student ID for some reason. Bring a California driver's license. A photo ID, we'll accept that instead. Okay. You don't need any notes, calculators, electronic devices, and it's really important that you don't answer your phone or pick up your notes or anything like that during the midterm for obvious reasons. Okay? Questions about the midterm or about anything else that you'd like to know about? You guys are so ready for this, right? All right. Yeah. Discussion on Friday. I think we need to, right? Because we're going to have we already have a new discussion worksheet that's posted, and we'll get too far behind if we don't have discussion on Friday.
This session for this week will that I know the only discussion that is recorded is tomorrow at 10.
Yeah, I hear you. Okay, so we posted the discussion worksheet, and it covers the material that's covered in today's lecture? So the answer is yes. Okay, but the worksheet and the key will be posted. Why don't we go ahead and post that today, okay? The key as well. Usually we post the key after the discussion. This week we'll post it before, okay? Is that all right? Okay. Other questions? Okay, again, if you want to know what's on the midterm, take a look at the practice midterm that I've already posted. Take a look at the problems in the book, the ones I've assigned in the book. Take a look at the worksheets, the discussion worksheets. When I go to write the exam, what I do is I sit down with all that stuff. And then I always have some concept problems as well, and I pull those straight out of the lecture. Okay, so the book is chock full of information. Okay, it's a textbook and it's written for a high-level audience. But you can focus down your studying just by focusing in on the stuff that's being covered and that I'm emphasizing by assigning it in problems. Okay, so that you don't necessarily have to memorize the whole book. Okay. All right, well, I wish you the best of luck. It's gonna be fun. You guys are gonna do really well, impress me. And I'm gonna be absolutely thrilled to see how well you guys do. Okay. Oh, okay, these are the announcements for this week. Start reading chapter 4, work the odd and asterisk problems. Again, the midterm this Thursday. Okay, so no other questions about the announcements, right? Last chance? Okay. So last time, what I was showing you is that DNA and circular plasmids can be folded to an astonishing degree. And I think all of us were blown away when we saw those smiley faces of DNA. That was plasmid DNA to which a large number of other single-stranded DNAs had been annealed to give you regions of double-stranded DNA that forced the plasmid into the happy face structure, okay, or the little map of the world structure. And that's pretty tour de force type of stuff. It's happening routinely in laboratories around the country, and it's something that you can sort of take for granted at this point. It's doable. We also talked about how you can use plasmid DNA to program cellular biosynthesis. And I told you about the requirements for plasmid DNA, that there has to be some selection marker and there has to be an origin of replication. Once you have those two requirements, you can encode all kinds of genes in that plasmid with all kinds of instructions. So the instructions could include things like, start building this factory that's going to produce diesel fuel, or start doing this particular function that causes you to glow green. Okay? So the ability to program cells using the plasmids has really taken off. And again, this is another technique that's sort of in the toolkit, and it's just commonly accepted as doable. And you can all simply start applying it on your proposals. Okay, now the other thing that we saw last time is that DNA polymerase is a remarkable machine. And note that it cranks out 1,000 covalent bonds per minute, not 1,000 per second. Okay, so it's really chirping along. That's a very, very fast speed at 1,000 per minute. And note that it's doing this with perfect fidelity, nearly perfect fidelity. It's one mistake every 10,000 or so. And that's truly remarkable. Okay, what else do I want to tell you about this? We talked about the model, the structural model, etc. Are there any questions about what we saw last time? Yeah, over here, Chelsea.
Question about the polymerase. You said it makes one error every 10,000. Does that include what corrects?
Yeah, so Chelsea's question is, does the one error every 10,000 or so, does that include error correction? And the answer is yes. So without error correction, its rate of errors would be even higher. But it's still really amazing. I mean, and also I should say this varies quite a bit depending upon which polymerase you're talking about. Okay, and reverse transcriptase is notable for having a much higher error rate. Okay, any other questions? That's a good one. All right, let's move then. Last time I ended up by showing you that you can manipulate the DNA of organisms as a way of studying the phenotypes of organisms. And on the last slide that we discussed, I showed you ways to randomly change DNA, and I'll talk some more about that in a moment. Before I do, though, I want to talk about how to actually program the DNA to have specific changes. Okay, so rather than going out and simply blasting the DNA and making changes here and there, which is kind of an unintellectual way to do it, a much more satisfying way would be to go in and spot weld in specific changes. Make a change here, make another change here, and have some hypothesis about those types of changes. It's a fundamentally different way of doing genetics than going for lots and lots of sort of random changes. Okay, so to make the changes specific, what people do is take advantage of the fact that you can cut DNA to have a little bit of an overhang, shown here in red, and then this overhang, if it has complementary Watson-Crick base pairing, that's A's and T's and G's and C's, with that complementarity, these two will come back together, and then you can use another enzyme called DNA ligase to finish up the last covalent bond in here where these arrows are. Okay, so simply by bringing together these two big pieces, they will find each other, form the perfect Watson-Crick base pairing, and then re-anneal. Incidentally, a technique not unlike this was used also for a lot of the DNA biotechnology that I showed with the folding of DNA. Remember where I showed, for example, patterns of DNA, not just the smiley faces, but other ones that involved a lot of these sort of techniques where you're cutting and pasting DNA. Okay, the result of this can be all kinds of interesting phenotypes, extra little bumps over here, frizzled little wings, so little shriveled up wings over here. And what's neat about this is that you have some hypothesis going into the experiment. You could say, I think that this particular gene is the gene that causes wings to extend out. And then you can test that hypothesis by disrupting the gene and seeing whether or not you get these little short shriveled, little shrimpy little wings. And you can use that sort of knowledge to confirm and potentially learn even more about how development works in other processes. Okay, so let's talk first about how to make DNA that has these overhangs. They're sometimes called sticky ends. Okay, so again, this is the overhang, also known as a sticky end. So to do that, we take advantage of a special pair of scissors, which, like normal scissors, has a similar sort of symmetry. This is what it looks like structurally. This little hollow region in here grabs onto the DNA and, like scissors, clamps down around it. When these cut, these class of enzymes are called restriction enzymes. When restriction enzymes cut DNA, they target palindromic sequences, specific palindromic sequences. Each restriction enzyme targets typically one and usually only one sequence of DNA. And note that this is a palindrome. You all know what palindromes are, right? Palindromes are sentences or phrases that are the same, that have meaning if they're in both directions, right? So for example, Napoleon was said to say when he arrived on the island of Elba, able was I ere I saw Elba. Okay, so do that backwards and you see able was I ere I saw Elba, right? Both ways. Okay, so that's a kind of a famous palindrome. Okay, madam, I am Adam, etc. Okay, so notice that these sequences are similarly palindromic, right? So this goes CAA, and then over here, okay, so it goes, let's see. So over here it's, let's see. Okay, so over here, GAA, so this is the palindrome, TTAA, and then over here, TTAA. Okay, so see there's a palindrome right there. It's the same going either backwards or forwards. Anyway, so these restriction enzymes have a symmetry to them, just like scissors, and that symmetry in turn dictates that they're going to be chopping apart palindromic sequences. When they make the cuts, these particular cutters are going to cut this bond over here and this other bond over here that are indicated by the arrows. And the result will be DNA that's separated and has a sticky end, two sticky ends, okay, by cutting apart these two different phosphodiester backbones. Phosphodiester bonds, this one and this one. Okay, so here's another picture of this. Okay, so here's the enzyme grabbed on. This is now a side view. The enzyme is grabbed onto the DNA. I've highlighted the DNA in purple and yellow to emphasize the two pieces that are going to come apart. And here it is, and it's double-stranded configuration. And then here it is after the restriction enzyme chops it apart. And the result now is a sticky end that will look for a complementary Watson-Crick base pairing sticky end. And when it finds it, it will then rehybridize, reform the Watson-Crick base pair, and you can then use this to glue together sequences of DNA. Okay, so typically the way this is done is you glue it back together and insert it into a plasmid that includes an antibiotic resistance marker. That's the selection marker that we talked about last Thursday, right? And I showed you a large number of examples of this. I showed you that you can use a plasmid that encodes beta-lactamase, chlorophetic-acetyltransferase, et cetera, tetracycline resistance, et cetera. Okay, so these sticky ends turn out to be very useful for pasting in new DNA. You can take advantage of that and do all kinds of things. You can insert in entirely new sequences in here that happen to have the perfect sticky ends, and then they will anneal, and so now you're spreading apart the yellow and purple pieces. Okay, so here's what this looks like in practice. Here's your plasmid. You chop this apart by a restriction enzyme. That gives you a plasmid that has two sticky ends. You then chemically synthesize foreign DNA, or maybe you get this foreign DNA by PCR or some other technique, and you set this up so that it has a nice sticky end hanging out over it, and then you can re-anneal this blue DNA together with the red plasmid DNA. And the last step here is an enzyme called DNA ligase that makes the last 5' phosphodiester bond to make perfectly covalent, closed circular plasmid DNA. Okay, and then you send this back into bacteria. Okay, and there's ways of punching holes in bacteria to get this plasmid DNA to flow in, and only the bacteria that take up the plasmid will be allowed to live. All the rest are going to die because you're going to treat the bacteria with some antibiotic, and only the ones that have this plasmid that encodes resistance to the antibiotic are allowed to survive those conditions. Make sense? Okay, yeah.
How do you keep the plasmid DNA from like re-annealing back onto itself? What's the efficient?
Okay, yeah. So that's a good question, and it's Anthony, right?
Yeah.
Okay, Anthony's question is a really good one, and it's one of those things that drives people nuts. Okay, his question is, what happens if the plasmid DNA re-anneals and closes back up? So this is a little bit technical, but what we do is we chop off the 3' hydroxyl, the 5' hydroxyl over here on this sticky end. Okay, so there's some 5' hydroxyl that's hanging off. Sorry, not hydroxyl, 5' phosphate off the 5' hydroxyl. We chop that off. So now it's just a hydroxyl, no phosphate, and then it can't reclose. The ligase can't operate on it. Okay, so that solves that problem. The problem is we still end up with some plasmid that comes through, and there's always some picking and sequencing and stuff like that. Okay, so let's talk about modifying proteins. Protein modification is a tool that's used very routinely in chemical biology laboratories. It's what, again, I encourage you to use in your proposals. Very, very straightforward. Okay, so the way we do this is we change the DNA sequence and then use those changes to the encoding DNA sequence to result in changes to the protein. And so one good way of doing this is something called quick change mutagenesis. This is actually spelled correctly. It's intensely annoying to me. A German company came up with this one. Anyway, so this quick change mutagenesis works by having new DNA sequences shown here in blue that encode some mutation. Okay, those are the X's. And then you use DNA polymerase to fill in the rest of the plasmid. So that's shown here in blue. Okay, and the tricky part is you then treat this resultant double-stranded DNA with a special restriction enzyme that operates only on methylated DNA. Okay, so DNA that's had methyl groups transferred to it. And do you remember earlier we talked a little bit about DNA methylation as a method for modifying DNA after synthesis. I just mentioned it in passing. But E. coli also do methylation. And so this means that this enzyme will chew apart the green strand of DNA and also the yellow strand of DNA from your original E. coli and leave intact the blue in vitro synthesized DNA. Okay, so the yellow and green is chewed apart by this special restriction enzyme, and the blue remains intact. And then, so what this means then is when you transfer it into the cells, a process called transformation, the cells then take up this blue DNA preferentially because this yellow and green stuff is chewed apart, and hopefully you'll get your mutation preferentially. And this actually works with very high efficiency. You can get, I would say, 50 to 90% efficiency out of this process pretty readily. Okay, any questions about how to make mutations? Okay, so everyone feels comfortable designing a mutagenesis experiment? Okay, well this is really powerful because now you can go in and test specific hypotheses. For example, in this protein over here, where in red, this is a heme cofactor, you could test the role of residues such as histidines that are interacting with this heme cofactor. So let's say you wanted to know, what is the imidazole functionality of this side chain contributing to the ability of this protein? You know, let's just say that this is some electron transfer protein, and you have some hypothesis that an aromatic ring in the protein is really key for transferring electrons here between point A and point B within the protein. You can then mutate the protein, replace the aromatic ring with just a methyl group. Okay, so you've now taken out that aromatic ring, and you can then test the variant proteins, the protein mutants, which we'll call variants, and test whether electrons can still move between point A and point B over here. That's really powerful. Okay, if it turns out the electrons still move, maybe your hypothesis that the aromatic ring is critical was wrong. Or maybe it turns out that they can't transfer anymore, and the aromatic ring is really crucial. So this allows you to do reverse engineering of proteins, right? You can take apart specific pieces of the complicated machine called a protein, and then examine what each one of those little parts does to contribute to protein functionality. Okay, any other questions about DNA biotechnology? Otherwise, I'm going to move on. Okay, there's a lot to talk about here. This is an absolutely fascinating field. It's one of those areas where there's sort of constant research activity. There's always new techniques that are coming along, and it's also tremendously fun as well. I would say this is really one of those sort of fun techniques. Like organic synthesis, you get to make stuff. It can be frustrating at times, but when it works, it really works well. Okay, I want to switch gears now, and I want to talk to you about DNA reactivity with small molecules. And we're going to start with the earliest type of reactivity of DNA, which is simply you walking around in the sun. Okay, so after this lecture is over, you start walking back to your dorm room, and although it's January, there's still a little bit of sun out there, and chances are you'll raise your eyes to the sky and be grateful you're living here in beautiful California. During that time, your face and your skin is being hit by UV radiation, and that UV radiation is causing damage to the DNA in your skin cells. Okay, and here's what's happening. If you happen to have two binding residues, two T's that are stacked on top of each other in a DNA sequence, these can do a 2+2 photocatalyzed cyclization to result in a cyclobutane structure, an adduct of your DNA. Okay. Again, this is a straightforward reaction, and I'll just very briefly show you the mechanism here. Okay, so it's two olefins, and you're adding light, which again, as usual, we abbreviate as H-nu, and the result here is a cyclobutane. Okay, straightforward mechanism, not particularly complicated. Here's the problem though. When this happens, this cyclobutane distorts the DNA. DNA no longer has this nice beef structure that we're so used to. And this is a problem. Okay, this is something that your cells have evolved to deal with. Okay, because organisms on this planet have always faced UV light. Okay, so even before we were destroying the ozone hole, there was still UV light that was sneaking through. And so a series of enzymes have evolved to fix this problem. And in goldfish and in rattlesnakes and other organisms that spend their lifetimes out in the sun, they actually have an enzyme called photolyase that reverses the 2+2 photocyclization that I'm depicting over here. Okay, and let me just make this arrow a little bit better because it's going to annoy me all lecture otherwise. Okay, so in this retro 2+2, this enzyme harnesses UV light and then pumps electrons into the cyclobutane adduct and breaks it apart. Okay, so the irony here is that it takes the same energy, energy from UV light, and this time uses that energy to reverse the reaction and drive it in the opposite direction. Now to do this, this enzyme has evolved antennas to capture that UV light. The enzyme has two antennas highlighted here in yellow and red, and I'll show you chemically the structures on the next slide. Okay, so one of these antennas is tuned to absorb the UV light and pass excited electrons to an FADH2 cofactor, which then pumps the electron into the cyclobutane. Let me show you what it looks like over here. So here's the antenna that absorbs the UV light. It's called MTHF. Its structure is shown here. The problem is, okay, so this works great. Okay, so the E. coli, the goldfish, the rattlesnakes, etc., they're fine. Okay, they can spend their days out in the sun. Us humans, though, we humans, however, have not evolved this enzyme. We don't have photolyase in our genome. We're not capable of running this reaction. Okay, and so what we do instead is we slather on sunscreen. Okay, that's what we've evolved to do in terms of we've evolved intelligence, and intelligence has come up with ways of dealing with this. And notice that the structures of sunscreen, this para-aminobenzoic acid, or PABA molecule, looks very similar to a key constituent of the UV-absorbing antenna used by photolyase. Okay, so again, we don't have DNA photolyase, and the sun causes the DNA damage I showed on the previous slide. So this PABA can, when it's on the surface of the skin, absorb the UV light and then radiate it out as simply heat. Okay, so the energy is converted from being electromagnetic energy that's going to excite electrons and cause the 2+2 photocyclization I showed, and instead it's going to be dissipated as heat instead. Problem is, over time, of course, PABA has absorbed enough UV energy that it's no longer so effective. It starts to break down. And so, and also the other problem is that the stuff is soluble in water and sweat. And so over time, it loses its effectiveness. And so you kind of have to be out there continually slathering yourself in this stuff. And no one is more expert in this than myself because of my skin coloring. And so, suntan lotion and I are very good friends. Okay. Now, let's talk about other fronts to DNA. Another problem with, that DNA encounters is that small molecules can also target DNA. And one example of this is a compound called sorolin, which is found in limes. Okay, and so, for example, it limes, the skin of the limes have this compound sorolin present in them. And I guess this is best dramatized by a group of school kids at a Baltimore day camp who are making pomander balls. Okay, and I bet no one in this audience knows does anyone know what a pomander ball is? Anyone? Okay, well, I'm not too surprised. I didn't know either. Pomander balls are these limes that have cloves stuck in them, and they're often wrapped with ribbon. So this is an example. Okay, so this is a lime, and then some little kid has stuffed a bunch of cloves onto the outside, and you're probably wondering, why would anyone want to do this? Okay, so evidently, people then throw those in with their underwear, in their underwear drawer, and it makes everything smell kind of nice, okay? That's the idea, at least. So anyway, a bunch of kids in Baltimore were at a day camp, and they were making these pomander balls using limes. And so they're rubbing this lime juice and lime oils on their skin. And what ended up happening was their skin broke out in all kinds of lesions, just sort of erupted in these terrible red lesions as the sorolin in the lime reacted with their DNA. I'll show you pictures in a moment, okay? But let's take a close look at the chemistry that's happening here. So what's happening is, actually this is predictable. Does anyone want to hazard a guess as to how this compound interacts with DNA? James? Intercalator, yes. Pi stacking, exactly. So this is one of these flat aromatic compounds that's going to slip into the pi stack of the DNA, right? Okay, so now imagine this. You're rubbing your skin with this stuff, and now it's slipping into your DNA. That's kind of scary, isn't it? Okay, and here's what it looks like structurally. So here's the sorolin. Here it is in the pi stack, and it has olefins perfectly positioned to react with DNA and form again those 2+2 after a photocatalyzed 2+2 cyclization reactions form these cyclobutanes. And again, the problem here is that the DNA is now cross-linked. It's distorted. It can no longer be used for replication. It could inappropriately cause transcription, right? Because now you have this hydrophobic mass in the middle. So when the transcription factor comes along and starts scanning the sequence of DNA, it gets all confused. And that's bad news. It can start inappropriately turning on genes that shouldn't be turned on, okay? And what happens is you get these terrible red rashes. Okay, so I have some graphic pictures in the next few slides. And if you're kind of a softie like me, turn your head away. Okay? But if on the other hand, you're planning to go to, I don't know, obstetrics or something like that, or you really like gory stuff, the next few slides are for you. Okay, I can't resist, all right? It's part of the fun part of learning about this stuff. Okay, so here's what it looks like. What it looks like is you get these sort of red rashes that look like that, and maybe this isn't so dramatic, but what ends up happening is on the skin, you basically can have lesions wherever it is that lime stuff comes in contact with your skin. Okay, so this happens a lot with farmers who are out picking limes or celery or other fruits or vegetables that have sorolins in them, right? Because that's the part that's exposed. It also happens to bartenders, right, who are mixing margaritas in the sun. But it also happens to college students, right? So if you happen to be doing belly shots, and here's the juice of the lime that's kind of like running across this person's abdomen, you end up with these bizarre streaks. Basically everywhere the sorolin managed to get to, and that can be a lot of places. So for this reason, college students are at particular risk for this. So that's one of the several PSA announcements that I'm going to have today. In fact, this whole lecture should be subtitled, How to Protect Your DNA. Okay, so number one, no belly shots out in the sun. Do them indoors. Let's talk about the processes that are enacted when DNA is damaged. Okay, so in order to divide, cells go through a complicated cycle. And it's a little bit like the military in that all cells have to either advance up the ranks or they're culled from the population. They must either advance or die. And so cells have a cell cycle. The timing of the cell cycle depends on the cell type. Some cells, as you know, are dividing much more rapidly than other cells. The cell cycle is depicted briefly here, and again, I'm giving you kind of a 10,000-foot view. The book provides more detail, and then other classes provide even more detail. Okay, so at some point in this cell cycle, the cells have to, in order to be able to divide, the cells go into a synthesis phase when DNA is being replicated. And during this process, there are a number of checkpoints and other sort of sensors that check to see whether or not the cell is ready to advance to the next stage, in this case the G2 stage of the cell cycle. And these checkpoints are highlighted by these red arrows over here. Okay, so many anti-cancer drugs target this S phase of the cell cycle. For example, you can imagine something like sorolin would massively disrupt this S phase of the cell cycle. The DNA is cross-linked. The two strands cannot be separated, which is a requirement for DNA replication. So let's talk about how many of these compounds work. I've shown you the 2+2 photocatalyzed cyclization. A more common mode for reaction with DNA involves using DNA simply as a big nucleophile. It turns out that the DNA bases are hugely nucleophilic. Okay, the backbone, negatively charged, not so nucleophilic, but the bases themselves, lots and lots of opportunities to act as a nucleophile. The reason for this is that there's lots of lone pairs that are orthogonal to these aromatic rings, that are sticking out from the aromatic rings, like this one over here on this adenine base. Okay, so where this arrow is pointing, there is a lone pair that is sticking straight out away from the ring, and that lone pair is not participating in aromaticity. It is totally ready and available to react with any electrophiles that it happens to encounter. So for this reason, many electrophiles are fantastic carcinogens. Okay, if we look at carcinogens as a class, they are largely electrophiles. There's some other classes as well, but in general, you want to avoid electrophiles if you can. Okay, and I'm going to show you some examples. Here's an example that I showed in an earlier slide. Okay, so in an earlier slide, this was from Thursday's lecture, when I showed you that you could treat fruit flies with random mutagens, and that would result in, I forget what it was, I think it was like extra eyes that were growing out of the fruit fly. The way that that reaction worked is, here is the nucleophile of DNA attacking the sigma star bond between this methyl group and this good leaving group of a sulfonate. Okay, and so what happens now is this DNA has been modified, right? You now have a new methyl group over here. And the problem with that is, now, instead of having a lone pair there that a transcription factor can recognize, now you have a hydrophobic functionality that might be going out to recruit transcription factors improperly. And so it might start turning on genes that otherwise might not get turned on. Okay? Yeah. Here's some examples. So here's again the structure of DNA. That's the B form of DNA. This is the major groove. This is the minor groove. The arrows are pointing to the most nucleophilic of the lone pairs. And all of the most nucleophilic lone pairs are lone pairs that are on aromatic rings but are not participating in aromaticity. Okay, notice that both of these rings have 6 pi electrons, 2, 4, 6, 2, 4, and then 6 in this lone pair over here. This lone pair, the one that has an arrow pointing to it, and this other one with the arrow pointing to it, don't participate in aromaticity. Okay, they're kind of spectators in the whole aromaticity business. And because they're not participating, they're really available to act as nucleophiles. They're super nucleophiles. The problem, of course, is that they also have a role to play in terms of encoding sequences, and that role can be messed up when they are modified. Okay, so it turns out, and I don't want to panic you too much, it turns out actually, even if you encounter a really large amount of electrophiles, you're not automatically going to get cancer. Okay, and there's actually an absolutely fascinating case study on this. In this case, let's see. In the case that we're going to see, there was an ex-boyfriend of this woman here who wanted to poison this family by giving them all cancer, and he unfortunately had access to chemical mutagens. He worked in a laboratory, I believe in Kansas, and he had access to this compound over here. And so he started, or actually, sorry, it's not this compound, it's another simple electrophile. And what he did was actually add large quantities of an electrophile to milk and to lemonade that he found in the refrigerator. Okay, so he stops by the house, he looks for an opportunity, goes to the refrigerator, and he dumps in large quantities of these chemical mutagens, shakes the stuff up, and then disappears. okay? And then later, when other people come to visit, she serves them this cold lemonade that has the mutagen. Okay. Oh, here it is. Here's a picture of him. This is Sherry's former boyfriend. Here's Sherry again. What he served, or what he, oh, here's a picture of his, disgusting little chamber of horrors. But what he did was he actually gave them these N-nitrosoamines, which are potent DNA alkylating agents. And why don't we take a look very briefly at the mechanism of this? Okay, so this is the nitrosoamine, and in the liver, it goes through an interesting activation step, where eventually it gives you this diazo compound shown here. Okay, notice that this has nitrogen as a leaving group. Okay, so earlier I showed you sulfonate as a leaving group. You can't get any better than nitrogen as a leaving group, right? This is N2, bubbles off as gas, fantastically stable leaving group. It does not get any better than this. Notice too that the electrons get to bounce their way over to a positively charged nitrogen. They love doing that. Okay, that's electron paradise. And so this makes us a very, very potent DNA alkylating agent. Okay, this is obviously something that you don't want to ingest. All right, let's get back to our story. What happened to the family over here? It turns out that they got, they died. Actually, the Dwayne Johnson and his infant nephew began bleeding inside and just basically died in tremendous agony. However, they didn't get cancer. Okay, so they died. It was horrible, but they did not get cancer. Okay, so none of the victims got cancer. Not all of the victims died. There's this fascinating book called Toxic Love that can tell you more about this. Here's what we learned from this episode. Okay, so first, DNA is a terrific nucleophile. On the other hand, it's very hard to convince the cells to induce cell death, and it's really hard to cause cancer. Even when you have enormous quantities of these known carcinogenic materials. Okay, so that there's enough sort of error correction in your DNA machinery and your cellular machinery to fix basic changes to the DNA, to fix modifications to the DNA. I'll show you that on the next couple of slides. This is great news. Okay, this means that you can all safely walk back to your dorm rooms knowing that your DNA is getting cross-linked by the 2+2 photocatalyzed cyclization that I'm showing over here, and also knowing that you're not going to die of that. Okay? So your cells are very good at fixing these types of affronts to the DNA. Okay, and in fact, actually, this stuff is totally ubiquitous in our daily lives. How many people had bacon for breakfast? All right, anyone else? Sausages? Anyone have sausage for breakfast? What do you guys eat for breakfast? Okay, well, at least one person had some bacon. I'm a big bacon fan myself. Here's what happens, though. The problem is that sodium nitrite is used as a preservative for bacon, for bologna, for sausages, etc. And the problem is that by heating and also other acid catalyzed reactions, which can happen in the stomach, this sodium nitrite can get rearranged to eventually form a terrific electrophile. Okay? So as the primary preservative in packaged meats, you get this fantastic electrophile. The good news is that this is probably not something that we have to spend a lot of time worrying about, because at the same time, vitamin C, which is also ubiquitous in our foods, can actually reduce this. It's an antioxidant, meaning it reduces things. It can reduce this electrophile, giving us a much more harmless molecule, nitric oxide. Okay, so here's ascorbic acid, that's vitamin C, and it can react very quickly with this reactive nitrite and prevent the formation of nitrosoamines. Okay, so here is the nitrosoamine over here, and for the reasons that are shown here, this is why you do not want to have nitrosoamines around. Again, this is a nitrosoamine. It eventually will rearrange to give you a great DNA alkylating agent, but vitamin C reacts with the nitrite, preventing the formation of those nitrosoamines. Okay, so the thing that I'm trying to tell you is that DNA will do a lot of reactions with a lot of molecules, But there's no reason to panic, okay? Because the chemistry on this stuff is remarkably complex. There's a lot of other molecules around. And furthermore, the cells have evolved really effective mechanisms for repairing damage to the DNA. And we'll take a look at that on a future slide. Before we do, let's take a look at some carcinogens that aren't found in our daily lives. Okay, at least I certainly hope not. Many of these are used to treat cancer, various kinds of cancer. Not so commonly now, but this was sort of our front lines in cancer, anti-cancer treatments some time ago. So these classes of compounds are known as DNA alkylating agents for the reason that these are electrophiles that react with DNA and leave the nucleophilic DNA alkylated. So they're going to come along and modify the DNA. Some of these are ones that are kind of familiar to us. Okay, so these nitrosoamines over here have a similar reactivity to the nitrosoamines that I showed a couple of slides ago. Others, like these nitrogen mustards, we'll talk some more about, okay, or the cyclophosphamide. Okay, so let's start at the top. Mesolates. Mesolates, great leaving groups. Okay, so this compound, busulfan, can alkylate the DNA. Notice that it has not one, but two mesolates. That's a problem, because that means then it can react with both strands of the DNA. And that's always bad news, right? Because now the DNA is cross-linked, right? It has the two strands that otherwise should be held together by hydrogen bonding are now covalently welded together permanently, or semi-permanently. Similarly, this bis-chloromethyl ether, also a potential cross-linking agent, right? Two leaving groups, two chlorides, which can act as leaving groups. Mom chloride, it has one leaving group. Okay, bad news, alkylates DNA, gets into cells very effectively. These are starting to look like compounds that you encounter in the laboratory, right? We routinely use, for example, chloroform, methylene chloride. We use lots of alkyl halides, for example. Okay, these are things that you encounter in insecticides. Let's see, methyl bromide, for example, is routinely used to treat grapes, and it's used for tenting houses. You know, when you see a tent over a house for termite control, that's actually, it's working using a DNA alkylating agent that's sprayed into the tent. Okay, these guys over here, the nitrogen mustards, we'll talk more about in a future slide. This one, again, is just like the nitrosoamines that I showed on the previous slide, which is to say this eventually rearranges to give you a diazo leaving group. Okay, now, here's the thing. Many of the compounds I showed on the previous slide are also used as anti-cancer drugs. And the idea there is that you're going to give these to patients and hope that the cancer cells, which are dividing very, very rapidly in the patient, more actively and more avidly take up the drugs than other normal cells in the patient. Okay, and for the most part, this works. Okay, I mean, it's true that these things are also incredibly toxic, and they'll, they'll kill other rapidly dividing cells in the poor patient. But at the same time, they are preferentially loaded into cancer cells. Okay, so the compounds I showed on the previous slide also appear in many anti-cancer compounds. Okay, so this is the dimesylate compound. This is a combination of a nitrogen mustard and also a nitrosourea. This is a nitrogen mustard. And these all have little niche markets. For example, this nitrogen mustard is used to treat a rare form of cancer that's found on the skin, specifically hands, and it's a tiny little market. It might be $50 million a year, but it's used as an ointment. And so patients who have this will get this ointment, rub it on their hands, specifically in the area, and use this to kill any rapidly dividing cells that happen to be in that area. Okay, and that's actually kind of nice because then it's specifically targeted just to that area as an ointment. Paracelsus, who is an absolutely fascinating character in the history of science, had this great quote that goes something like, and I won't read it to you in the German because my German's not up to it. It basically says, show me any compound and I'll show you two sides to it. One side, it's a toxin. The other side, it can be a treatment, a cure. All this depends on the dose. Okay, so at high doses, this can be a toxin. At low doses, these things could be used as treatments. Okay, that's sort of the paradox of these things. So the goal of chemotherapy is to induce cell-programmed suicide called apoptosis in rapidly dividing cells faster than you induce new cancer, new carcinogenicity in normal cells. Okay, that's the big challenge for anti-cancer treatments, at least up until the last, I'd say, 15 to 20 years ago. Okay, and in the last 15 or 20 years, chemists have started coming up with compounds that are much more specific at targeting what makes cancer cells cancer cells. Okay, the compounds I'm talking to you about today are ones that simply obliterate any DNA that they happen to find. And the future really is targeting the specific attributes of cancer that makes that cell a cancer. Okay. So let's talk a little bit about how your cells repair themselves after an affront. You're walking home and you decide to chat with your friend out in the sun, and so you end up with some cross-linking of thymines. Okay, this is the photocatalyzed 2+2 that I'm showing over here. Okay, so the way this works is that your cells have a constellation, a fleet of DNA repair proteins that are constantly circulating. Okay, the analogy would be tow trucks that are driving around on the 405 looking for broken down cars that are disrupting traffic, and then as soon as they find one, they just pull it off the road rather than disrupting traffic. Okay, so you have these DNA repair enzymes that are constantly scanning your DNA, looking for things like cyclobutane adducts, okay, looking for this sort of affront. When they find this, they don't simply snip out the mistake over here. Instead, what they do is they snip out a big segment of DNA, a big chunk, 10 base pairs on this side, 10 base pairs on that side. And this part is excised using a restriction enzyme. And DNA polymerase comes along and fills in the correct sequence of DNA. Okay? So it's an excision repair mechanism, okay, that you chop out a big chunk and then DNA polymerase moves in. The reason you can't simply remove just those couple of bases is that DNA polymerase likes to get a running start. Okay, it doesn't work so well just on a couple of bases. It really needs 20 or something before it can start cranking along. Just doesn't, it's a professional. It doesn't like to do small jobs. Okay. Any questions on what we're seeing? All right. Okay, so monofunctional alkylating agents, things like this mom chloride compound that I showed on a previous slide, are relatively harmless. Okay, they're not super lethal because they can be so readily fixed. This is not to say that you should go out and start inhaling mom chloride. Okay, you want to stay as far away from that stuff as possible. Here's the mechanism. In this mom chloride, what's happening is you're actually forming an oxonium ion intermediate, and this oxonium ion intermediate is a fabulous electrophile. Right? Notice that the oxygen bears a positive charge, and we know oxygen, by virtue of its position on the periodic table, does not like having positive charge. It's electronegative. And so this makes this an exceptional DNA alkylating agent. And here it is attacking or being attacked by the nucleophilic DNA. And again, this can be readily fixed using excision repair. Okay, the problem is if you have too many changes, okay, you can actually end up overwhelming the repair system. So if there are too many places that are alkylated, then it's just hard for the DNA repair machinery to keep up. It's just too much to handle. The other problem is that some DNA alkylating agents hit not one, but two strands of DNA. And this excision repair mechanism assumes that you have a second strand of DNA that's available to act as the perfect copy and to provide a template to fix the broken strand. What happens if both the strands have been damaged, say by X-rays or something like that? You're in trouble. That's a real problem. Okay, so bifunctional cross-linking agents are insanely toxic. Busulfane is a lot more lethal and damaging to DNA than two equivalents of methyl mesylate. the monofunctional DNA alkylating agent that we've been seeing today. Okay, so cross-linking DNA, far more damaging than monofunctional adducts. And again, that's because the cross-linking can hold together the two strands of DNA and prevent you from having the template strand to act as a copy during DNA repair. Okay, so here's a couple of examples of this. And this is, in this example, this is a nitrosourea. What's important about this is it gives us, again, the diazo leaving group. So that's a nitrogen as a leaving group, this diazo compound, analogous to what we saw with the other nitrosoureas. But then this other chloride can hang along, form this adduct of DNA over here, and this second strand of DNA can attack this. And this gives you now two strands of DNA bridged by an ethyl functionality. Okay, so now they're covalently held together. And again, this is bad news. Okay, excision to repair works pretty well, except when it encounters these double-stranded breaks. These sort of double-strand where you get cross-linking or double-strand breaks. For that matter, excision repair can also be lethal if it's not, if there's a genetic abnormality that encodes a protein that's no longer functional. Okay, and there's actually a fantastic movie about this called The Others and it includes a great performance by Nicole Kidman. Has anyone seen this? Okay, spoiler alert. The kids have this disease in which they have abnormal DNA repair enzymes. Okay, if you haven't seen this, you should still see it. I haven't totally ruined the movie for you. Come close, but not totally. Okay, so missing or deficient DNA repair enzymes causes severe disease. Okay, we have a kind of a graphic image on the next slide. The disease xeroderma pigmentosa, for example, is caused by a incorrectly encoded DNA repair enzyme. And this poor girl has this. And the problem is, again, you're constantly being confronted with damage to your DNA. Even when you're outside in the sun, UV light causes the DNA to be cross-linked by this photocatalyzed 2+2 photocyclization. You absolutely require functional excision repair enzymes. Without those, you cannot live on our planet. Okay, there's just too much damage going on. And otherwise you end up with this terrible disease. Okay, let's get back to the story with the crazy guy, the psychopath who tried to murder the family by giving them all cancer. How come none of them died of cancer? It turns out that when we look at cancer cells, we find hundreds, if not thousands, of mutations to the DNA. Okay, so when we start sequencing tumors, we find a tremendously heterogeneous mixture of mutations, where each tumor is slightly different. And furthermore, when we take biopsies in different spots of this same tumor, we also find tremendous differences amongst those cells, which otherwise superficially look identical. Okay, so to form a cancer requires hundreds, if not thousands, of mutations to the DNA. It takes an enormous change to the DNA to do that. Furthermore, those mutations have to target 3 different control aspects of the cell. And I like to think of these as an accelerator, a clutch, and the brakes. Okay, I'm a car guy, so for me this analogy works really well. If you don't drive stick shift, you might not know that back in the day when all cars were manual, they had a clutch. The clutch was this extra pedal that you would push down to switch gears. Okay? And earlier I showed you that cells have to progress through a cell cycle and that there were checkpoints through that cell cycle. So I like to think of those checkpoints as shifting gears. Okay, so if the cell cannot shift between different phases of the cell cycle, it cannot proceed. However, if you have mutations to those checkpoints, to the proteins that are controlling the test for whether or not the cell should be allowed to progress, you can end up with mutations that allow the cell to inappropriately progress in the cell cycle, allowing runaway cell division. Okay, so those are mutations to the clutch, which are the checkpoint proteins, often kinases. In addition, all cells have on the cell surface growth factors and growth factor receptors which are responsive to the external environment. Mutations to these growth factor pathways are absolutely required. That's like a mutation to the accelerator. These growth factors are telling the cell, start dividing, start producing this, start doing this, get going on this. So that's the mutation to the accelerator. And finally, all cells have tumor suppressor genes. And we'll look in a moment at one called P53, which I like to think of as the brakes. This is the machinery that shuts down the cell when it starts running out of control. And when this happens, these tumor suppressors can trigger apoptosis, cell suicide that prevents cancer. Okay, so these hundreds to thousands of mutations have to affect all three of these pathways. If just one is affected, then there's a good chance that the cell will default into apoptosis and arrest and prevent the cancer. If, on the other hand, you have mutations to all three, then pathways can start running out of control. Okay, so let's see. If we take a very brief look at P53, which is the tumor suppressor protein, here's the cell, here's the self-destruct button, and there are a whole series of different questions the cell is constantly asking before it can go into cell division. And at any given time, if answer to any of these questions, is it big enough? Is there enough room? Are there 2 copies of DNA? Is the DNA properly lined up? If the answer is no, the cell will be immediately sent into apoptosis, where it basically blebs apart. Bleb is a fancy word for explode. Okay, it starts bleeding out these blobs off the surface. Bleb and blob are 2 great words. Okay, they sound like each other because that's really what's going on here. So P53 is a hair trigger sensor that can turn on apoptosis, and mutations to the P53 gene allow DNA damage to accumulate. So mutations to P53 are bound in like 60% of colon cancers, for example. This is a very, very common set of mutations that are behind a large number of different cancer types. And in fact, it's almost mandatory for some cancers to get going. Okay, because a functional P53 will shut down most of the runaway cell division that would otherwise cause cancer. Okay, so for this reason, things that you do that affects the P53 gene are really, really serious. And for example, scientists have been able to link mutations to P53 directly from smoking. Okay, so if you're out smoking, there's a good chance that you're starting to cause mutations to P53. This is literally the smoking gun. Okay, this is what causes the link between cancer and smoking. Which until this was done in the mid-90s or so, cancer companies, cigarette companies were able to promote their cigarettes as saying there is no definitive evidence that cancer causes smoking. Here is the definitive evidence. What happens is you end up with mutations to the residues that are highlighted in yellow. So these are residues that have a positive charge and interact with double-stranded DNA. Notice that P53 does a lot of things in the cell. One thing it does, quite importantly, is act as a transcription factor to trigger apoptosis. And if these positively charged residues are mutated, the P53 can no longer bind to the DNA, and it can no longer trigger apoptosis. So smoking affects P53. It introduces mutations specifically at the molecular level to these residues here, and that in turn results in cancer. Okay? If you're smoking now, you should stop. Okay? If you know people who are smoking, you should persuade them to stop. I cannot think of anything worse. My father died of lung cancer. I am passionate about this. Stop smoking now. I mean, you might as well just put your mouth up to like the exhaust pipe on a bus or something. Okay? It's as crazy as that. All right. Now, what is it about smoking that causes cancer? We actually know quite a bit about why it is that unburnt things cause cancer, and it goes back a long ways and a long history of biology. And I guess we have to go back to the father of epidemiology, the great Sir Percival Pott. Percival Pott, shown here, was a physician in London, and he noticed that his chimney sweeps had a ridiculously high level of testicular cancer. Okay, so chimney sweeps are these guys who in 18th century London would actually climb into chimneys. Okay, so this isn't Santa Claus. This is actually, you know, real humans that are climbing into the chimneys as a way of cleaning them out. And they would carry brushes that look like this, and they would just be covered in soot. Okay, because they're in the chimney where there's all this sort of unburnt sooty stuff. In the unburnt sooty stuff, what we find are chemicals that cause testicular cancer and other types of cancer. And I have a picture of testicular cancer on the next slide. Sorry, I couldn't resist. If you have an aversion to this sort of thing, avert your eyes. I'm convinced, though, by showing you these images, that at least one of my students someday is going to benefit from this. I'm really hoping that I'll prevent at least one person from, or maybe I'll have helped one person detect cancer very early. Okay, so here's a picture of a cross-section through a testicle. Check out the size over here. This is in centimeters. So this whole region in here is one giant cancer that has grown. So you end up with these, you know, just tremendous growths of cancer in testicles. Now here's the good news. Good news is, if it's caught early, this is totally treatable. Okay, this is the sort of thing that if you catch it early, it can be stopped by both chemotherapy and also surgery. Okay, and so men in the audience, you should all be thinking about monthly self-check exams and get more information here. And again, this is another one of my PSA announcements. I really hope someday I'll have prevented someone from dying. That would be the coolest thing. Okay, so testicular cancer. Oh, okay, another picture. Okay, this one's a little bit gorier. These are actually the cancers highlighted over here. A little hard to see in there, but again, this is totally treatable. Okay, so Percival Pott is noticing that in London, a very high percentage of his chimney sweeps are coming down with cancer, testicular cancer, and he's wondering, what is it about the soot? He hypothesizes that the soot is what's causing this testicular cancer in chimney sweeps. We now know that in unburned carbon or unburned stuff, there are a lot of carcinogens, and I want to show you that on the next slide. Okay, so this benzopyrene is found in high concentrations in unburnt carbon. Okay, so question over here. Oh, let's see. That was a picture of an operation to remove cancers. So, best not to look at it too closely. We can talk more about it later. Go C-section, okay? All right, so these compounds over here are definitively cause cancer. Okay, so for example, if you take benzopyrene and you rub it on the backsides of these rats, they come down with these horrible cancerous lesions, those bumps over here. And these work by mechanisms that you can predict, right? These are flat aromatic compounds. How do they work? Flat aromatic? Yes, on three. Let's do it together. 1, 2, 3. Intercalators, yes. So these flat aromatic compounds fit into the DNA. They slide straight into the pi stack. But in addition, they also can alkylate the DNA. And this is less obvious, and I want to show you this on the next slide. Okay, so when you smoke cigarettes, or for that matter, when you smoke alternative things, there are unburnt bits in there. And those unburnt bits are benzopyrene. Okay, and the benzopyrene that gets into your liver is epoxidized. Okay, so it's, your liver will try to process this stuff. Okay, so you can imagine this stuff is not very soluble in water, and it's really bad news to have insoluble fragments of stuff floating around your bloodstream. So the liver does its very best to deal with this, and the liver's strategy for solubilizing insoluble matter is to oxidize it and introduce hydrophilic functionality that would make it soluble in water. Okay, and so here is an enzyme in your liver oxidizing the benzopyrene. Here's a successful oxidation to make a diol. Here's a second oxidation that creates, instead of a diol, an epoxide. Epoxide has a strained three-membered ring. This strained three-membered ring is a fantastic electrophile. Okay, so now this is really bad news. You now have this this intercalator that's sliding into your pi stack that has the perfect alkylating agent, electrophile, delivered right up against your nucleophilic DNA. And that's really the problem. Okay? This is an enormous problem. Okay? What ends up happening is the DNA gets modified covalently by this benzopyrene. Okay, so for this reason, countries that eat a lot of barbecued food, that have a lot of sort of burnt stuff in their diets, tend to come down with high levels of stomach cancers. Okay, likely because they're counting benzopyrenes in their diet. Your goal should be to try to eliminate benzopyrenes as much as you possibly can. There's lots of stuff out there that people are paranoid about that they think is bad for them. Here's one that we genuinely know is bad for you, and here's something that you can do to help yourself live a lot longer simply by avoiding it. All right, here's another one. This is aflatoxin. Aflatoxin is produced by molds that grow on grains like peanuts. Okay, and in the United States, all of the peanut butter is tested for the presence of this mold, this Aspergillus flavus mold. But in other countries where the public health systems aren't and the sort of food safety mechanisms aren't as vigorous, this is more of a problem. But the problem is the mold looks like this, and it gives off this aflatoxin, which again is modified by cytochrome P450. I should have called it. This is cytochrome P450 in the liver, and that introduces an epoxide. This epoxide is a fantastic electrophile for nucleophilic attack of the DNA. And once the DNA is modified, this eventually leads down to DNA strand cleavage. Okay, so modified DNA eventually leads to DNA that's been chopped apart. That DNA that's been chopped apart is no longer available, and if you get hundreds of these mutations, eventually you get cancer. Okay, bad news. All right, let's talk about the nitrogen bustards. Okay, so earlier I showed you compounds that had a nitrogen, ethyl group, and a chlorine. Here's a variant where it has a sulfur instead of a nitrogen. Okay, in both of these compounds, they both go through a common mechanism, which is the heteroatom in the center of the compound can act as a nucleophile to form a terrific electrophile. And in this case, the nucleophile attacks and is alkylated. If this nucleophile is DNA, then the DNA can be cross-linked, giving you two strands that are covalently welded together. Okay, so earlier I showed you, for example, I believe it was busulfan, the compound I told you about that you rub on your skin for this very rare case type of cancer. It's used as a chemotherapeutic. These were also used as war gases in World War I, which is completely insane. Okay, that you would actually do this to anybody on the planet. But in any case, these compounds cause cancer by forming fantastic electrophiles, which then react with the nucleophiles found in the DNA. Okay, so again, here is the nitrogen mustard equivalent, and these are compounds that we saw earlier in today's lecture. And all of them go through a common intermediate, this aziridinium ion over here. And notice what a great electrophile this is, right? The nitrogen has positive charge on it, and nitrogen hates having positive charge. So when the nucleophilic DNA attacks here, the electrons get to bounce their way to the positively charged nitrogen, setting up sort of the perfect, perfect electrophile for modifying DNA. Okay. Any questions about the nitrogen mustards, the sulfur mustards, DNA alkylating agents? Okay. I want to switch gears now, and I'm going to take the last 5 minutes to start talking a little bit about RNA, just to kind of whet your appetite as we talk about RNA. Okay, there are no other questions about DNA, right? Questions about cancer, anything like that? So RNA, chemical biologists have come around to recognize RNA's tremendous importance in cell biology. As an example of this, this is actually a structure of DNA that was posited by Phoebus Levine, who I'm going to show on the next slide. And you know, this is around like 1920 or so, and structure of DNA and RNA was not very well understood. Okay, so he posited that this DNA structure was the structure of DNA. Of course, this is totally wrong, right? We know what the correct structure of DNA is. But what Levine did right is he also assigned pentose ribose sugars in nucleic acids. And he specifically showed there was deoxyribose in DNA, and also figured out that DNA is a nucleoside to denote the glycosidic bond. Okay, so it's possible in science to be totally wrong, yet get some details correct. Okay, so this structure to us looks totally nuts, right? It's only 4 bases of DNA arranged in a circle. But on the other hand, when you look more closely at the details here, you start to find all kinds of interesting features. Like, for example, that the bases are held on by glycosidic bonds, that this has a phosphodiester backbone, that the connectivity here is 5' to 3', etc. Okay? So in a similar way, we chemists have had sort of a reevaluation of the importance of RNA. When it was originally discovered, it was thought to be merely a transfer, sort of a go-between between DNA and proteins. And in the last 20 years, our appreciation of RNA's key role in a cell has expanded enormously. Okay? So we now know that RNA can act as a soldier, sailor, and tinker, and spy. And first, as a soldier, RNA is actually a very effective catalyst for cleaving its own sequences. It can actually go out and cleave sequences of other RNA strands. As a sailor, transfer RNA delivers amino acids to the ribosome. Here's transfer RNA delivering amino acyl tRNAs to the ribosome during protein synthesis. As a tinker, the ribosomal RNA can act as a catalytic machine to synthesize proteins. And finally, as a spy, messenger RNA encodes proteins. So RNA is capable of all kinds of things. And this means that when we come back on Tuesday, we're going to have a lot to talk about. And I'll look forward to that then. Thanks.